in addition to the statistical significance of the fact that this cross on the left typically produced a 2-to-1 ratio of yellow to non-yellow, the authors also discovered that when they set crosses between two yellow mice, shown here on the left, the brood sizes were often smaller, in this case a total of 6 individuals, than crosses where they had a yellow mouse mated with a wild-type mouse, in which case those brood sizes tended to be larger. And that was another clue about what was happening and why the authors were not observing a Mendelian ratio of phenotypes in the yellow cross. We now know that the parental genotypes in this cross are the yellow mouse is heterozygous, with the capital Y allele being dominant and producing yellow coat colors in the offspring that contain the capital Y allele. And the wild-type mouse is homozygous recessive. So in this case, all of the offspring that are produced from this cross, if they're yellow, they get the capital Y from the parent that's yellow, and they have to get the lowercase y from the wild-type mouse, the parent on the left. So in this case, the Punnett square reveals that the yellow mouse has capital Y and lowercase y alleles, and the wild-type mouse produces only lowercase y. So when we combine those two alleles at fertilization, producing the F1s, those are the genotype ratios, 1-to-1, heterozygous to homozygous. So this cross works as Mendel would have predicted. Another point to make is that the alleles, the number of capital Y's and lowercase y's, is in what we call Hardy-Weinberg equilibrium. And this is based on a pair of scientists who identified that when nothing is going wrong with a cross, when one allele isn't better than another allele, when they're both equally useful, equally viable, equally healthy, the number of capital Y and lowercase y alleles won't change from generation to generation. So for example, here's the P0 generation, here's the F1 generation. And let's count the number of capital Y alleles and lowercase y alleles in the two generations. So in the P0 generation, there's 1 capital Y and 3 lowercase y. So 1 capital Y, 3 lowercase y. Now let's do the same thing for the F1 generation. There are 1, 2, 3, 4 capital Y's, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 lowercase y's. And you'll notice that the ratio is the same. For every 1 y, capital Y, there are 3 lowercase y's. The same ratio here, 4/12, 1/3. The number, the ratio, the frequency of capital Y's to lowercase y's doesn't change in a population from generation to generation if neither allele is better or worse than the other one. That is called Hardy-Weinberg equilibrium. The equilibrium indicating that there's no change expected to occur. Restated, this cross exhibits Hardy-Weinberg equilibrium, and the genotype ratios that are produced are the ones that you would predict based on Mendelian segregation patterns. That is, the heterozygote genotype produces yellow mice, and the homozygous recessive phenotype produces wild-type mice. So there's a 1-to-1 genotype ratio in this cross, and that corresponds to a 1-to-1 phenotype ratio. Now let's take a look at that yellow by yellow cross and see how it differs. So here again is that cross between two yellow parents that produce 6 offspring in that 2-to-1 phenotype ratio. And the genotypes of these parents are both heterozygous. So, what does the Punnett square look like for this cross? Each parent will produce 50% gametes carrying a big Y and a little y. So when we combine those gametes at fertilization in all possible random combinations, assuming each combination is equally likely, then we see that there are 3 different possible genotypes: homozygous capital Y, heterozygous, and homozygous lowercase y. And there are 2 different ways to get the heterozygous condition. So for every 1 homozygote, there are going to be 2 heterozygote individuals for every 1 homozygous recessive individual. And that's what Mendelian inheritance would predict. Now let's look at what the genotypes of the F1 actually are in this cross. It turns out that all of the offspring from this cross, the yellow ones, are all heterozygotes, capital Y over lowercase y. And of course, the only way to get the original wild-type coat color back is those are the homozygous recessive individuals. That is, the only individuals being produced in a cross between 2 yellow mice are those that are homozygotes for the wild-type allele and heterozygotes. And what's missing are individuals that are homozygous for the yellow mutation that causes the yellow coat color. So it turns out what's happening here is that individuals that are homozygous for this mutation that causes yellow coat color are lethal. Those individuals die before birth, so they never exist for us to observe and to genotype them to find out what their genotype is. If we look at the phenotype ratio from this 1-to-2-to-1 genotype ratio, all of the individuals that have 1 or 2 capital Y alleles should be yellow. So that's 3 to every 1 individual that should be wild type. So what we should observe, and what we predicted based on Mendelian inheritance, was a 3-to-1 inheritance pattern. And that's what our chi-square test ruled out. Instead, we saw a 2-to-1 inheritance pattern. And that's because of these offspring, 2/3 of them, or 2 out of 3, do have a capital Y allele, and the other third, 2 out of 6, are homozygous recessive. This is a great example of how Mendelian inheritance, although expected, doesn't actually occur when one genotype is less healthy than other genotypes in the same generation. And this we can observe by performing the same calculation, tallying allele frequencies between the parental generation and the surviving offspring generation. So again, for this cross, in the P0 generation and the F1 generation, let's tally the number of capital Y alleles and the number of lowercase y alleles. So again, in the P0 generation, we have an equal number of capital and lowercase y's at 2 each. And in this case, we have 1, 2, 3, 4 capital Y's in the second generation, the F1s. And we have 1, 2, 3, 4, 5, 6, 7, 8 lowercase y's. This is a change in allele frequency. We've gone from equal representation of the two alleles to an overrepresentation of one of the two alleles, twice as many in this case, 8 to 4, in the second generation. This is evidence that Hardy-Weinberg equilibrium is not occurring. And the way that geneticists interpret this is that means that one allele appears to be better or healthier or more fit than the other, and that would be the one that's overrepresented in the second generation. This is, in other words, evidence that there's something wrong with some individuals, specifically that carry the capital Y allele, that they are less frequent in subsequent generations. So to reiterate, the reason that Mendelian ratios aren't observed in this yellow cross is because some combinations of alleles are lethal. Capital Y homozygotes are not surviving to be counted in offspring litters. So in our lab class then, what we're going to be doing is something very similar. We're going to be looking at crosses, and we're going to be genotyping the existing individuals, not the ones that die or are lethal, to see if some genotypes are missing. So in this case, again, that offspring generation was totally missing one possible combination of parental alleles, capital Y over capital Y. So, to find out if a gene is inherited in the expected fashion, we look in the generation to see if the ratio of genotypes is 1 to 2 to 1. And if one genotype's missing, that genotype might participate in lethality. If that genotype's not missing, like it was in the Y over Y, capital Y over capital Y case, if it's just underrepresented, if there are fewer of that genotype than expected, that could still be an indication that that allele participates in some sort of non-lethal incompatibility. And that's what we're going to be looking for. Deviations from Mendelian expectations can give geneticists a clue about the presence and location of genetic incompatibilities.